Good morning, everybody. Can you hear me? Good
Good morning.
morning. Can you hear me?
Yes, Doctor, I can.
Okay. And you can see the screen also.
Yes.
Okay. Thank you. So we'll start today. So this session is a continuation of previous session where we did some part of the development of the nervous system. So here we are going to go a little bit on details because now you have learned most of the neuroanatomy and you know the different structures of the central nervous system. So initially, if you remember that we, in the first session when we started with the development of the nervous system, we were talking about the vesicles. So from the neural tube, we talked about the three vesicles, the prosencephalon, the mesencephalon, and the rhombencephalon. And the lower part, the caudal part of the neural tube, give rise to the spinal cord. But now what we would do is that we will go into details of these three vesicles, the mesencephalon, rhombencephalon, and prosencephalon, and then see how different structures, both gray matter and white matter structures, come out from these, the three vesicles of the brain, which were formed in the neural tube. And in the final part piece of this session would be, some of the congenital abnormalities which we haven't touched before. So the purpose of, or the main objective of this session is to describe the development of myelencephalon, which is the lowermost part of the rhombencephalon, the metencephalon. So myelin and metencephalon are parts of the rhombencephalon, or the lower or caudal most part of the vesicles. And then we are going to talk about how diencephalon forms and what structures come out from diencephalon and telencephalon, and then relate the common congenital abnormalities associated with these parts of the neural tube. So we have distinct basal plates and alar plates representing the sensory motor areas are found on the each side of the midline in rhombencephalon. So if you see, this is the prosencephalon, this is the mesencephalon, and this is the rhombencephalon. And this flexure which you are seeing is the pontine flexure. And this pontine flexure divides the rhombencephalon into cranial piece, which is the metencephalon, and then you have the caudal piece, which is the myelencephalon. Myelencephalon is going to give rise to the medulla. Metencephalon is going to give rise to the pons as well as the cerebellum. Mesencephalon gives rise to the midbrain, and the prosencephalon, which it was, one unit, one vesicle, it gives two outpouchings, which are called the telencephalon, and the centerpiece of the prosencephalon is called the diencephalon. So telencephalon, which are the two outpouchings from the prosencephalon, is going to give rise to the cerebellum, while the centerpiece of the prosencephalon, which forms the diencephalon, has a roof part and a basilar part. The roof part is thalamus. The basilar part is going to give rise to the hypothalamus. But coming back to the structure, we saw that you have the basal plates in the spinal cord lying ventrally, and the alar plates of in the spinal cord lying dorsally. And you know that basal plates give rise to the motor neurons, and the alar plates in the spinal cord used to give rise to the sensory neurons. Now, in the brainstem, this arrangement changes, and this basal plates, which were lying in front, they come in the center, while the alar plate goes to the sides. So laterally located are alar plates, medially located are the basal plates. And basal plates are going to give rise to the motor neurons, and the alar plates are going to give rise to the sensory neurons. So this is what is written here, that you have distinct basal and alar plates representing motor and sensory neurons, and they are found on each side of the midline. So near the midline, you have the basal plate and the motor nuclei, and laterally you have the alar plates, which are going to be the sensory neurons. In prosencephalon, however, you would not have basal plates, you would have only the alar plates. So the neurons which are seen in prosencephalon, that means T-encephalon and diencephalon, they are basically coming out from the alar plates, and basal plates in the prosencephalon regress. So now we are going to put our focus on the rhombencephalon. And among the rhombencephalon, we are looking at the caudal part of the rhombencephalon, which is the myelencephalon. So myelencephalon extends from the pontine flexure to rhombocephalic isthmus. So this is the rhombocephalic isthmus. This is the pontine flexure. So this area is called myelencephalon, and this myelencephalon give rise to the medulla oblongata. So how this myelencephalon, which is the vesicle which we are talking about here, extending from the pontine flexure all the way to the rhomboisthmic isthmus, rhombocephalic isthmus. This part is the myelencephalon, which gives rise to the medulla. Now, if you look at the spinal cord, as I said, you had basal plates located ventrally and the alar plates located dorsally. So your motor neurons, the anterior gray horn cells in the spinal cord were lying in front, while the sensory neurons or the dorsal gray horn cells were lying posteriorly. Now in the medulla, what happens is that the basal plates are going to lie medially, and you would see the alar plates lying more laterally. And they are separated by a sulcus, which is called the sulcus limitans. So you see, this is the sulcus limitans here, which is dividing the floor of the medulla into two pieces. You have the medial portion, which has basal plates. You have lateral portion, which has the sensory or the alar plates. Now, the basal plate has three set of nuclei. The medial most part of this basal plate has the somatic efferents, which are the nuclei which supply the muscles which have originated from somites. And in the case of medulla, this is basically the muscles which are the muscles of the tongue. So this is the hypoglossal nucleus, which is lying much more medially in the basal plate. Then next to it is the special visceral efferent, and these are basically the branchial efferent. These are the muscles which have originated from the branchial arches. And in the case of medulla, this is the muscles of pharynx and larynx, and the nucleus which we talked about is nucleus ambiguous. But it doesn't stay here, it migrates into the reticular formation of the medulla, and that's why this is called nucleus ambiguous. You cannot actually make the location of this nucleus. The third piece of the basal plate in the medulla is the lateral, general visceral efferent, which is the parasympathetic nucleus. And the parasympathetic nucleus in the medulla is the dorsal vagal nucleus, which supplies the smooth muscles in the lungs, in the heart, cardiac muscles in the heart, and then smooth muscles in the intestine. Uh, Myelencephalon, if in the myelencephalon, the column which is represented by neurons, we have already talked about the three groups. Then we come to the alar plates, and the alar plates has three groups of sensory nuclei. The lateral most, which you see here, is the somatic afferent sensory group. It receives the impulses from the trigeminal nerve as well as the vestibulocochlear nerve. Then you have the intermediate or the special visceral efferents. They receive impulses from the taste buds of the tongue, from the palate, and from the oropharynx. And then the most medial is the general visceral efferent group, which receives the pain and temperature as well as interoceptive impulses or information from the GI tract, like distension or sensory information, general sensory information from GI tract and the heart. So these are the three new set of nuclei which are related with the alloplates. The roof of the myelencephalon. So this is on the floor of the myelencephalon. The roof of the myelencephalon is basically formed by a single layer of the ependymal cells, which is covered by a vascular mesenchyme, forming the pia mater and choroid plexus of the fourth ventricle. So what you are seeing basically is that the floor is formed medially by the basal plate and laterally by the alar plate, and the roof of the myelencephalon is formed by ependymal cells covered by vascular parenchyma and the pia mater. The two combined structures, the pia mater and the vascular mesenchyme, is called the tela choroidi, which is continuously producing the CSF. It forms the choroid plexus and it forms the CSF. Now we are coming to the metencephalon, which is part of the rhombencephalon, the cranial part of the rhombencephalon, just above the pontine flexure. Um, and metencephalon is also characterized by both basal and alar plates, so similar to the arrangement which you have seen in the myelencephalon, that medially there is basal plate, and laterally his alar plate. Medial part or the basal plate is going to give rise to the motor neurons, and there are subset of motor neurons which you are going to see, and the alar plate is going to give rise to the sensory set of neurons. There are two components which are going to get out from metencephalon. One is pons, and the other is the cerebellum. So the origination of cerebellum also happens from the metencephalon. So as we said, first we can look at the pons, and we can just see the structures which will come out from the basal plate and alar plates, and then we will go and see the development of the cerebellum. So from the basal plate, again, you have the motor neurons coming out and forming the motor neurons of the pons in this case. So medial group is always in the basal plate is the medial somatic efferent, which is basically the muscles which have originated from somites. And in the case of the pons, you know that there is only one muscle which is supplied, and that is the muscle of the eye, which is the lateral rectus muscle, and it is supplied by the sixth cranial nerve, which is obtusent. The special visceral efferents, which supply the muscles which have originated from the branchial arches, you would find them at two levels: at the lower level of pons as well as the upper level of pons. At the lower level of pons, they are going to form the motor nucleus of the facial nerve and supply muscles of facial expression. At the upper level of pons, they are the motor nucleus of the trigeminal nerve and supplies the muscles of mastication. So special visceral efferent or branchial efferent, which is basically in the second column which is going to arise from the basal plate, at the lower level of pons is going to be the muscles of facial expression and the nucleus of the facial nerve, while at the upper level of pons, the same column of nuclei is going to supply the muscles of mastication, and it is the motor nucleus of the trigeminal nerve. Then the third column on the basal plate is going to be the general visceral efferent, which is the parasympathetic nucleus. And the parasympathetic nucleus in the pons is the the superior salivatory nucleus, and this superior salivatory nucleus supplies the submandibular, sublingual, and the lacrimal gland, and it is basically the parasympathetic nucleus of the facial nerve. Okay. Uh, in case of the in case of the pons, there is something which is very special here. What you see is that you the marginal layer, you have alar and basilar plates, but the white matter is the marginal layer. So in the case of metencephalon, the marginal layer expands and it forms the bundles of fibers which cross the ventral part of the pons and then go to the opposite cerebellar hemisphere, forming the bridge. In the pons also, you would get in the ventral part the pontine nuclei, which originate from the alar plates of the metencephalon. So these pontine nuclei, which you see here in the ventral part of the pons, they originate from the alar plates and move forward as the marginal layer of the in the marginal layer in the metencephalon is going to expand, forming the transverse fibers which are crossing the pons ventrally and going to the opposite cerebellar hemisphere. There would be certain cells from the alar plate which is going to migrate into this marginal layer, forming the pontine nuclei. Now we come to the alar plates. Again, in the alar plates are mainly going to give rise to the sensory neurons, and there would be three groups. The lateral group in the lower part is going to give rise to the vestibular complex, which is related to hearing and balance. And in the upper part of the pons, the same lateral somatic afferent group is going to form the principal sensory nucleus of the trigeminal, which is important in terms of receiving the sensory information from the face and the head and neck region. Then the second group of the sensory nuclei in the alar plate are the special visceral afferent group. We don't have only in the lower part of the pons, you have the uppermost most part of the nucleus solitarius, which is going to be for the taste sensations from the anterior two-thirds of the tongue. General visceral efferents are usually not represented in the metencephalon. Now we are coming to the development of the cerebellum, and we said that the cerebellum also arises from the metencephalon. So if you look at the posterior part or the dorsal view of the metencephalon, you would find the lips, some of the caudal portions of the metencephalon is giving an extension which is called the rhombic lip. Can you see this rhombic lips here? So the dorsolateral part of the alar plates of the metencephalon are medially extended, and as they are medially extended, they are forming the rhombic lips. Cranially, these rhombic lips are joined together, and as the pontine flexure is formed, more and more pontine flexure is formed, this rhombic lip is going to form a plate-like structure, because as the pons is going to flex more and more, there is more pontine flexure. The two lips, the rhombic lips, are going to come together and they are going to form a plate, which is called the cerebellar plate. So around 12th week of intrauterine life, you would start to see this cerebellar plate, and this cerebellar plate shows a central portion, which is the vermis, and the two lateral portions, which are the cerebellar hemisphere. And then a transfer fissure soon would separate the nodule from the vermis and form the lateral flocculus or flocculonodular lobe, which is the primitive lobe which you are seeing here. So you see this is a transverse fissure which is going to give rise to this flocculonodular lobe here, and it will separate the cerebellar hemisphere from this flocculonodular lobe, which is going to be lying here. So this transverse fissure, which you see here, is going to separate the cerebellar hemispheres from the flocculonodular lobe. Uh, Initially, the cerebellar plate consists of the neuroepithelial cells, the mental layer, and the marginal layer, as you saw in the spinal cord. But during further development, what happens is that the neuroepithelial cells, which have migrated to the surface of the cerebellum, they form an external granular layer. These cells keep on dividing in the proliferative zone of the cerebellum, and ultimately this external granular layer is going to give rise to the three layers of the cerebellar cortex, which you see, which include the molecular layer, the pyramidal layer, the Purkinje cell layer, and the granule cell layer. So this external granular layer, which has originated from the neuroepithelial cells, is going to differentiate as we, come close to the birth into three layers of the cerebellar cortex, which you see, the molecular, Purkinje, and then granule cell layer. And within the white matter, you would see the deep cerebellar nuclei. So what is happening is that the neuroepithelial cells migrate to the surface of the cerebellum, forming the external granular layer. This external granular layer keep on, proliferating, and then it will result in the formation of the definitive cortex, which include the molecular layer, Purkinje cell layer, granule cell layer, and within the white matter, there would be the deep cerebellar nuclei. So this is a description given here. So we can see that during later stages, cells of external granular layer migrate inward to mingle with the Purkinje cells and form definitive cortex of the cerebellum. The dentate nuclei or the deep cerebellar nuclei is one of these deep cerebellar nuclei, and it is going to be originated from the same, external granular cell layer. So in the 6th month of the development, the external granular layer gives rise to the various cell types, and these cells migrate towards the differentiating Purkinje cells and give rise to the granule cells. Basket and the stellate cells, which you see in the cerebellar cortex, are produced by the proliferating cells in the cerebellar white matter. And the cortex of the cerebellum, consisting of Purkinje cells, Golgi 2 neurons, neurons produced by the external granular layer, and they reach their definitive size at birth. Uh, and the position, the deep cerebellar nuclei, such as dentate nuclei, they also reach their final position before birth. So you can see that around 6 months of intrauterine life, the external granular layer start to differentiate into different cellular types of the cerebellar cortex, and the other cell types like basket cell and stellate cells, they are produced by the cerebellar white matter, not from the external granule cells. And then you can see that the definitive size of the cerebellar cortex is almost reached around birth, and also the deep cerebellar nuclei are going to reach to their definitive position around birth. Now we're coming to the midbrain, which is the mesencephalon. So once we finish the rhombencephalon, the vesicle, which is just above the rhombencephalon or the cranial to the rhombencephalon, is mesencephalon, and mesencephalon is going to give rise to the midbrain. Again, you have the basal plates and the alar plates. The basal plates you can see in front. Now here they have only two sets of or two groups of neurons in the which originate from the basal plate. One is the medial somatic group, and this medial somatic group is going to supply the muscles which have originated from the somites, and these are the muscles of the eye which move the eye. And these are at the lower part of the midbrain. This is the trochlear nucleus, which supplies only one muscle, which is superior oblique. In the upper part of the midbrain, there is the oculomotor nucleus, which is going to supply all other muscles of extraocular muscles of the eye, which move the eye. Then next to the somatic efferent group, we have the general visceral efferent group, which is the parasympathetic nucleus. And this parasympathetic nucleus here is the Edinger-Westphal nucleus, which supplies the smooth muscles of the eye, the sphincter pupillae, and the the ciliary muscles. The marginal layer of the basal plate enlarges as it enlarges in the mesencephalon. Similarly, the marginal layer of the basal plate is going to enlarge here to give rise to the crus cerebri. So the crus cerebri, which you see where you would find that there are the corticopontine fibers and the corticospinal and corticonuclear fiber, they originate from the marginal layer of the basal plates of the mesencephalon. Initially, the alar plates, which you see at the back, they are divided into two elevations, a superior elevation and a lower elevation. The superior elevation is going to give rise to the superior colliculus, which is important in the visual pathway, and then the lower elevation is going to give rise to the inferior colliculus, which is important it's an important auditory relay area or the nuclei. So the alar plates in the mesencephalon are going to divide into two superior elevations and two inferior elevations. The two superior elevations are the superior colliculi related to the visual reflexes, and the two inferior elevations are the inferior colliculi, which are important in terms of the auditory relay stations. Prosencephalon, we said, the uppermost part vesicle which you saw is going to give rise to two output chains, which is going to give rise to the telencephalon, which give rise to the cerebrum, and the center part of the prosencephalon is the diencephalon, which forms the optic cup, the stalk, the pituitary, the thalamus, the hypothalamus, and the epiphysis. So the diencephalon is going to give rise, the central piece of the prosencephalon, which is the diencephalon, mainly going to give rise to hypothalamus below and thalamus up, but it also gives rise to other parts of the central nervous system, including part of the pituitary. It is going to give rise to the optic cup, and it is also going to give rise to the give developmental origination to epiphysis. So the roof plate of the diencephalon, which is the central piece here. So the roof plate of the diencephalon, and it gives rise to the posteriormost part of the roof plate is going to give rise to the epiphysis. The most of the roof plate of the diencephalon is going to form the choroid plexus of the third ventricle. So the diencephalon, which develops from the median portion of the prosencephalon, is thought to consist of the roof plate and two alar plates. So the roof plate of the diencephalon consists of a single layer of the ependymal cells covered by the vascular mesenchyme. Together, these are going to give rise to the choroid plexus of the third ventricle. And the most caudal part of the roof plate of the diencephalon, because we know that in prosencephalon there is no basal plate. You have two alar plates and you have a roof plate. So the roof plate in terms of the diencephalon is forming the choroid plexus mostly of the third ventricle. The most caudal part of the roof plate is going to give rise to the epiphysis. The two alar plates which you see in the diencephalon, the cranial part of it is going to give rise to the thalamus. The caudal part of it is going to give rise to the hypothalamus. So you see here, this is the alar plate, And the lower part of this alar plate, or the ventral part of this alar plate, is going to give rise to the hypothalamus, the mammillary body, the infundibulum, which is the diverticulum, which participates in the formation of the pituitary gland, because pituitary gland is formed by two parts: the infundibulum, which is the diverticulum from hypothalamus, and Rathke's pouch, which is coming from the oral cavity, from the ectoderm around the oral cavity, and both of them are going to join to give rise to the pituitary. Rathke's pouch going to give rise to the anterior pituitary. The infundibulum is going to give rise to the part of the posterior pituitary. But this is the alar plate of the prosencephalon. The roof of the prosencephalon, or the roof of the diencephalon, is going to form, or roof plate is going to form the choroid plexus. Posterior most part is going to give rise to the epiphysis, while the alar plate itself laterally, the ventral part of it, the dorsal part of it is going to give rise to the thalamus. The ventral part of it is going to give rise to the hypothalamus, mammillary body, and infundibulum. Again, you are seeing here, this is the roof plate, and this is the alar plate which you are looking at. The roof plate is going to give rise to the choroid plexus of the third ventricle, and the caudal most piece is going to give rise to the epiphysis, which is going to give rise to the pineal gland and structures around the pineal gland. And this is the lateral part, the two alar plates. And within the alar plates, the ventral portion is going to give rise to the hypothalamus, mammillary body, infundibulum, while the dorsal part of the alar plate is going to give rise to the thalamus. So alar plates of the diencephalon, which is the middle piece of the prosencephalon, is going to give rise to the thalamus and hypothalamus. Alar plates forms the lateral wall of the diencephalon. So lateral wall of the center part is the third ventricle. Lateral piece of the diencephalon is formed by the alar plates of the diencephalon. The groove which separates the thalamus and hypothalamus is called hypothalamic sulcus, which divides the plate into dorsal portion, which is the thalamus, and the ventral portion, which is the hypothalamus. One other thing which comes out from this alar plate is the mammillary body, and also you see this infundibulum coming out from the floor of the hypothalamus, and that is going to give rise to the posterior pituitary. We have already talked about the development of pituitary, both Rathke's pouch, which is the ectoderm around the stomodium, and infundibulum, which is the projection from the floor of the hypothalamus. Rathke's pouch is going to give rise to the anterior pituitary, while the diverticulum from the floor of the hypothalamus is going to give rise to the posterior pituitary. Now we are coming to the some of the clinical correlates. There might be some hypophyseal defects. For example, occasionally small portion of the Rathke's pouch persist in the roof of the pharynx, and this is called the pharyngeal hypophysis. Then you have the craniopharyngiomas, which arise from the remnants of the Rathke's pouch. These are tumors which arise from the remnants of the Rathke's pouch, and they may form within the sella turcica or along the stalk of the pituitary, but usually lie above the sella turcica. They may cause hydrocephalus, they may cause pituitary dysfunction like diabetes insipidus and growth failure. Now we are coming to the last piece of the development. One is the formation of Diencephalon. So Diencephalon is the most rostral part of the brain vesicle. So prosencephalon is going to give rise to two outpouchings, and these outpouchings are called the telencephalon. and they are going to give rise to the cerebral hemispheres. And the cavity of these cerebral hemispheres is called the lateral ventricle. And this lateral ventricle is going to communicate with the third ventricle through the foramen of Monro. So telencephalon, the only thing which you need to remember, telencephalon is the outpouching of the prosencephalon. And these outpouching, lateral outpouchings are going to give rise to the cerebral hemispheres. The cavity of the telencephalon are the lateral ventricles. And these lateral ventricles are going to communicate with the cavity of the diencephalon, which is the third ventricle, through the foramen of Monro. By the middle and the second month of the basal part of the hemisphere is going to, extend uh, forward over the thalamus and begin to grow and bulge. And so initially they are lateral outpouching, but then they extend forward and beyond the thalamus, giving rise to the frontal lobe, and then back of the thalamus, giving rise to the occipital lobe, and then below the thalamus to give rise to the temporal lobe of the cerebral cortex. If you take a transverse section, there are within the white matter also some deep-seated nuclei which you are going to see, which are the basal nuclei or the basal ganglia, which is developing within the cerebral cortex. So some of the congenital defects, one of the congenital defect which you can see, which is very, very rare, is called holoprosencephaly. Now, holoprosencephaly refers to spectrum of abnormality where you lose midline structures and results in the malformation of brain as well as the face. So in severe cases, you would not have two cerebral hemisphere, you would have one. So you would have one lateral ventricle rather than two lateral ventricles. You would have eyes fused in the center, so you have one eye, and there is a single nasal chamber rather than two nasal chambers which you see. So there are midline, fusion of the facial structures, and there is also fusion of brain structure. So that abnormality is called holoprosencephaly. Then there is another very rare type of congenital abnormality, which is called Scheisencephaly. And in this, there are large clefts which occur in the cerebral hemisphere. So you see normal gyri sulci, if there is large cleft in the cerebral hemisphere, then it is called Scheisencephaly. And sometimes it may cause loss of brain tissue. Then you have meningocele, meningoencephalocele, and meningohydroencephalocele. And we have talked about this. Meningocele can only, shouldn't be thought that meningocele is in the spinal cord. You can have the bulging of the meninges over the top of the brain. That can be meningocele. If there is brain tissue extending with it, then this is meningoencephalocele. And then if you have the ventricular cavity also extending, then it is called meningohydroencephalocele. So it can happen with the spinal cord, and it can happen over the skull also, that you have only meninges bulging, then it is meningocele. If it is meninges with the with the part of the brain bulging out, that is meningoencephalocele. And if the ventricular cavity is also extending, then the defect is called meningohydroencephalocele. Uh, so you can have part of the brain, you can have part only meninges bulging, you can have part of the brain bulging, and you can have a part of the brain plus the ventricle bulging. These defects you can see in 1 in 2,000 births, so they are not that rare as you see Scheisencephaly and holoprosencephaly, which are very, very rare congenital defects. So this is what is you are seeing. You are seeing only the meninges. There is a defect in the skull, and you have only the meninges bulging. This is the meningocele. If there is meninges, which are seen in the green, and then you have the brain tissue bulging, this is the meningoencephalocele. And if you have the meninges, the brain, and the lumen of the ventricle bulging out from a defect in the skull, then it is called meningohydroencephalocele. The other defects which you can see, one of the other rare type of defect is exencephaly, which is characterized by failure of the cephalic part of the neural tube to close. So if you have the cephalic part of the cranial tube to close, then you may have the brain tissue. There is no wall of the skull which is going to develop, and the brain tissue would be exposed to the exterior. There would be no meninges covering, there is no skull covering, and the brain tissue is exposed to the exterior. And later, this tissue is going to degenerate, giving rise to a large necrotic tissue. And the defect is called the anencephaly. When you see the necrotic brain tissue, exposed to the exterior, then the defect is called anencephaly. Anencephaly, although the brainstem remains intact, so the brain stem has nothing to do, but the the upper surface of the brain, because there is no skull, gets exposed to the exterior and gets degenerated. And you would see the necrotic tissue, exposed to the exterior. Since the fetus lacks the mechanisms for swallowing, the last 2 months of pregnancy are characterized by hydromenos. So with congenital abnormalities like exencephaly and anencephaly, the mechanism of swallowing is mainly affected, and because the fetus cannot swallow the amniotic fluid, usually it is characterized by polyhydramnios. The abnormality can be recognized on radiograph since the vault of the skull is absent. And anencephaly is common abnormality. It is present in 1 into 1500 live births. 4 times it is much more common in females than males. And like spina bifida, it can be easily prevented by giving 400 micrograms of folic acid per day before and during pregnancy. Then we are coming to some of the common congenital defects, and one of the common congenital defect is hydrocephalus, which is characterized by an abnormal accumulation of the cerebrospinal fluid within the ventricular system. And in most cases, the hydrocephalus which you see in the newborn, it is because of the obstruction to the aqueduct of Sylvius. So if the aqueduct of Sylvius is obstructed, the CSF cannot pass from the third ventricle to the fourth ventricle. And if it cannot come to the fourth ventricle, it cannot go into the to the subarachnoid space and then go over the top of the brain to get absorbed through the arachnoid villi. So this is more of the obstructive type of hydrocephalus. So this prevents the cerebrospinal fluid to move from the lateral ventricle and third ventricle into the fourth ventricle, and from there to go to the subarachnoid space to be reabsorbed. Uh, since the cranial structures have not fused at the time of birth, what you would see that the head, circumference will expand, and in extreme cases, the brain tissue and bones become thin, and the head may be very, very large. Then we are coming to the Arnold-Chiari malformation, which is the caudal displacement and herniation of the cerebellar structures through the foramen magnum. So the tonsils of the cerebellum, sometimes they pass through the foramen magnum, and this is basically associated with the spina bifida cystica, where there is tethering of the spinal cord with the vertebral column. And as the vertebral column is going to expand, it is going to pull the spinal cord, making the tonsils of the cerebellum to pass through the foramen magnum, and that is called Arnold-Chiari malformation or caudal displacement of the cerebellar structures through the foramen magnum. That can also obstruct the flow of the CSF through the roof of the fourth ventricle into the subarachnoid space, causing hydrocephalus. So Arnold-Chiari malformation usually is associated with the spina bifida cystica, with teething of the spinal cord with the vertebral column, and it is associated with the herniation of tonsils through the foramen magnum. Then we have something which is called microcephaly, which describes the cranial wall that is smaller than normal. And since the size of the cranium depends on the growth of the brain, the underlying defect is in the brain development. And the causative abnormality is varied, and it may be genetic, like autosomal recessive, or it can be due to prenatal insults like infection or exposure to drugs or exposure to teratogens. One of the common infections which can lead to microcephaly is the fetal infection by toxoplasmosis. So if you have a fetal infection during intrauterine life with toxoplasmosis, that can also lead to microcephaly. Leads to cerebral calcification, mental retardation, hydrocephalus, or microcephaly. These are some of the issues which you see with toxoplasmosis infection of the mother during with a pregnant mother, which results in the insults and leading to microcephaly in newborns. But you may also find, great many other defects of the central nervous system, which will show, you do a MRI or CT scan of the brain, and you would find absolutely no abnormality, and still there would be severe mental retardation, or there would be severe issues in terms of the brain development. On the other hand, you may find that, corpus callosum is completely absent. You may find complete absence of the cerebellum, and you would still find very little, gross abnormality in terms of motor sensory system or intellectual ability. So you is still we have not really come up with these congenital abnormalities, because sometimes we see absolutely normal morphology of the brain which is associated with severe mental retardation, or in the other hand, we see that there is gross abnormality like the absence of corpus callosum or the absence of the cerebellum, and we see very little abnormality in sensory, motor, or higher mental functions. So it's not necessary that you would see a structural abnormality in the brain which would be associated with mental retardation or defects in higher mental function or defects in sensory and motor function. You might see completely normal brain, but you still have mental retardation or abnormalities with the brain function. The leading cause of mental retardation is one of the leading cause of the mental retardation is the maternal alcohol abuse, and child born with mothers with a history of maternal alcohol abuse, they might have morphologically normal brains, but they may represent with severe mental retardation. Thank you very much. If you have any questions, I can answer them. Let's see the charts. So transverse fibers from the basal plates. Yes, for in the pons, in the metaencephalon, from the basal plates, the fibers are going to give rise to the transverse fibers of the which you see in the ventral part of the pons. Okay, thank you very much. Can you please repeat the beginning of the development of the cerebellum? Okay, so the the if you see the caudal and lateral part of the alar plates of the mesencephalon, they have, you know, a little bit of overgrowth, which are called the rhombic lips. These rhombic lips, as there is a pontine flexure formed, these rhombic lips are going to come together and form a plate, which is called the cerebellar plate. The center portion of this cerebellar plate is going to form the vermis. The lateral portion is going to give rise to the cerebellar hemisphere. So mainly, cerebellum is going to arise from the lateral portions of the alar plates of the mesencephalon, where you have the rhombic lip. And these rhombic lips, because of the pontine flexure, these rhombic lips, which are like this, they will move like this and form a plate. The central portion is going to give rise to the vermis. The lateral portion is going to give rise to the cerebellar hemisphere. Is it okay? So in the marginal layer of the pons are going to be expanded, from the basal plate, and it is going to give rise to the transverse fibers. Thank you very much. If there are no more questions, we'll stop here.